So the basic idea is that some microbes require oxygen to live, just like humans. So if you took human physiology recently, you probably learned a lot about cellular respiration and how oxygen is important for converting food energy into ATP energy. And a lot of microbes use that same energy strategy. They require oxygen just like humans. But some microbes have different ways of converting energy, and some of those are actually killed by the presence of oxygen. So they only live in environments that don't have oxygen in them. So for example, the intestines, the human intestines, there's not oxygen in there. And so the organisms that live deep in our intestines, a lot of them are killed if they become exposed to oxygen. They're perfectly happy living inside us with no oxygen. There's another group of microbes that don't care about oxygen. It doesn't kill them. They're fine with the presence of oxygen, but they don't use it for their energy. So they can grow in the presence of oxygen, but they're not using it for anything. The fourth group, there are some microbes that use oxygen, and that's their preferred way of getting energy, is using oxygen for cellular respiration. But they have some backup strategies so that if oxygen gets low, they can still continue to live, and they use a different energy conversion strategy. And then the last one there are some microbes that need oxygen, but they need a lower concentration of oxygen than what's found in the normal atmosphere. So normally the air that we breathe now, it's 21% oxygen. And there are some microbes that don't do well in that 21% oxygen. They need reduced oxygen, maybe like 5% oxygen instead. So they're super picky. Okay, so the way this assay works is we'll put the bacteria into a tube that has nutrients in it. So this is a broth that has nutrients so the bacteria can grow. And it also has a little bit of agar in it that makes the broth thicker than regular broth. And this little pink stripe up here is showing us where the oxygen is. This is just a chemical indicator that reacts with oxygen and turns pink. So the oxygen from the atmosphere can diffuse very slowly into this tube. It doesn't make it down very far though, because the agar is too thick. So just the top zone here has oxygen. The bottom part doesn't have oxygen. And so when we put the bacteria in here, we'll inoculate them all the way down to the bottom. Then the bacteria will grow in a pattern that lets us know what oxygen they prefer. So this first one here is bacteria that don't use oxygen, but they're not hurt by oxygen, right? So it's that first bullet point from the last slide. And growth we can see as cloudiness. Do you see how it looks all cloudy now? That cloudiness shows us that they're just growing everywhere. They can grow up here where there's oxygen. They can grow up here where there's not oxygen. They don't seem to have a preference for growing with or without the oxygen. They just don't care about the oxygen. The second tube here are ones that prefer to grow in the oxygen. So most of the cloudiness is up here, but they can grow down here in the not oxygen area. So you see this sort of streak coming through here. This is where they were inoculated, where the bacteria were added. And so it's a little cloudy down here, but they're mostly growing up here. This is the third type where they aren't growing up here in the oxygen. You see how we can still see it's really red up here. The oxygen hasn't been used up and there's no cloudiness, but the cloudiness is down here where they're growing in the low oxygen condition. These ones, it's just cloudy at the top. There's nothing down here. And so this is showing it that these bacteria have to have oxygen. They can't grow without it. And then the last one here is a little hard to see, but you see those little pink stripe and then a stripe of cloudiness underneath it. These are the ones who need low concentration oxygen in order to grow. They're not growing down here. They're not growing at the very top. They're just growing in this cloudy stripe here. Okay, so these are all the possible results that we might see on Friday when we come back. Today, what I'm going to do is just inoculate the bacteria into the tubes. I have 3 types of bacteria that I'm going to inoculate. We'll let them grow in the incubator, and then we'll come back and see the results on Friday. Here are my thioglycolate tubes. So I have 4 tubes and 3 kinds of bacteria. One of my tubes is going to be a control. I'm going to write C for control. And then the other tubes, I'm just labeling with the initial for the type of bacteria, AFSE. And instead of doing Clostridium butyricum, which it says in your lab manual, we're using Sporanges. It's just a related species that we had. But otherwise, it's the same as what's in your lab manual. All I'm going to do is use my loop to use aseptic technique to transfer a loop full of AF bacteria into the AF tube, a loop full of SE bacteria into the SE tube, and a loop full of CS bacteria into the CS tube. I won't touch the control, that will stay uninoculated, and these will go into the incubator. All right, flaming my tube, my loop nice and hot again. Flame the top of my tube, I'm waiting my 30 seconds. Okay. Getting a loop full of bacteria. And for this, I'm just going to go straight through the center down to the very bottom so it gets inoculated to the bottom and pull it back out. And I'm leaving the screw cap a little bit loose so that oxygen can get in and continue to diffuse down through the auger. Okay, so that's it. It's just, moving things from point A to point B. I'll do that for the other tubes.